and I'll do the electron transport system in a separate slide in a second. Okay, now, so you're starting off with your pyruvic acid. Now, do you remember we would have used, we would have used PACO to try and remember the different molecules that are involved in Krebs cycle. So so now what we're going to do is we're going to produce acetyl, and you need the name of this, acetyl coenzyme A, and then this now is going to go into Krebs cycle, the acetyl coenzyme A. So P, pyruvic acid, A, acetyl coenzyme A, and then we had these molecules. Now, the old course, they used to have to know the names of these, but you don't anymore. So you can just call C, K, and O. And I think that helps you there because, like, the pyruvic acid is a 3-carbon molecule. Okay, acetyl coenzyme A is 2-carboned. Okay, 6-carbon, 5-carbon, and 4-carbon. So it kind of helps you to remember where you're going to lose carbon dioxide in a second. So PACO helps with that. Now, the other thing here now about the pyruvic acid, it only enters mitochondria if there is oxygen present. Okay, so location of stage 2 or Krebs cycle, mitochondria, the lumen of the mitochondria, and pyruvic acid will only go into the lumen of the mitochondria if there's oxygen present. So oxygen is definitely required for Krebs cycle, and large amounts of energy are going to be released here. Okay, now so pyruvic acid is broken down to acetyl coenzyme A. I've already said that pyruvic acid is a 3-carbon molecule and acetyl coenzyme A is a 2-carbon. So you must lose a molecule of carbon dioxide. Any time that you go from a 3-carbon to a 2-carbon, well, where is the carbon atom going? From 3-carbon pyruvic acid to a 2-carbon to acetyl coenzyme A, it's going as your carbon in your carbon dioxide. So if they might say in the exam questions, what small molecule is released? Carbon dioxide is the small molecule that's released. And we breathe that out as it's a waste product of respiration for animals. So we breathe that out through our mouths and our noses. Now, we also produce NADH here. So just remember that, okay? So we make some NADH, and we make some carbon dioxide when you're converting pyruvic acid to acetyl coenzyme A. Okay, and then what happens is the 4-carbon combines with the 2-carbon to make your 6-carbon, C we'll call it here. Now look what's happening here, going from 6-carbon to a 5-carbon K, again, more CO2 is going to be released. And when you're going from 5 carbons to 4 carbons, more CO2 is going to be released. So you talk about the release of carbon dioxide there. And the other thing you're going to have to try and remember is that you have more NADH produced there. And you have ATP here, and you also have NADH. Okay, now this is all in your notes. You need to go over it. I'm just quickly revising it here for you. So what's going on in Krebs cycle is that the acetyl coenzyme A is being broken down. And remember, when you break down molecules, the bonds are broken in the molecule, and you're going to release energy. So you're breaking down acetyl coenzyme A to carbon dioxide and protons there, the H pluses that are producing the NADH, and you're also releasing high energy electrons as well when you're breaking down the acetyl coenzyme A. So the acetyl coenzyme A is broken down to carbon dioxide, high energy electrons and protons. The high energy electrons and protons go to the NAD plus, okay? Actually, I might just for a second there before I go on to the electron transport system, at the start, when we spoke about the energy carriers, they might ask you for the function of ATP. So if they do, you're going to say, what does it do? What's its role, actually, is the word they'll probably use in the exam question. So its role is it traps and transfers energy if they ask you for ATP. If they ask you for the role of NAD plus or NADH for that matter, the role there is that it traps high energy electrons and protons or H plus ions, okay? So NAD plus traps high energy electrons and protons. ATP traps traps energy or stores energy, if you like. So that's just if they ask you for the role of those. Now, finally then, again, just before we move away from Krebs cycle, remember that table that you would have in your notebook now. So location of Krebs cycle, the lumen of the mitochondria, requirement for Krebs cycle, well, you need pyruvic acid, but you also need oxygen there. Where do we get that requirement? Well, pyruvic acid you get from glycolysis, and oxygen you get from breathing it in the air. The product then of Krebs cycle, what 3 things did you make? Okay, well, in Krebs cycle you made ATP, okay, you made NADH, and you also would have made carbon dioxide, and you also need to know the fate of those three products. So the fate of ATP is it's broken down and it's used to provide or to release, whatever word you want to use, energy for what? Give some, it's for any metabolic reaction that happens in your cell. We possibly, when we were doing this, might have written the word aron next to ATP in your table so that we'd remember the characteristics of life. So if you're a living organism, you require energy for metabolic reactions, okay? So to be able to produce urine, to be able to reproduce, to be able to respire, to organize yourselves into cells, tissues, organs, functions, and that they function properly. So you would need muscular activity, nervous activity for nutrition so that you can feed. All of those, you need energy for any metabolic reaction, mitosis, anything. So the role of ATP or the function of ATP is to provide, to break down and to provide or to release energy. Remember we said in fixed or manageable quantities for immediate use within your cell, okay? The role of NADH then, you're just going to say there, like we said earlier, that it traps high energy electrons and protons for the electron transport system. So these will be taken to the electron transport system, and the carbon dioxide we exhale through our lungs. Finally then, the electron transport system, we would have said that it's made up of a series of molecules, or the molecules would be the steps, and what happens is that the NADH here breaks down to give you back your NAD plus, 2 electrons and protons. These 2 electrons, high energy electrons, and these are passed from molecule to molecule, and when they do, remember we talked about them being like hot potatoes, that they're passed very quickly from molecule to molecule, and these electrons release energy, which we can use then to produce ATP from ADP into P. So what will happen now though, remember, is that you've lots of low energy electrons here sort of gathering. So we talked about having to mop these up. Remember, what will mop them up? Oxygen. Really important that you mentioned the oxygen there. And you need the equation there too, I would say. So half O2, okay, these low energy electrons that you're trying to get rid of, so 2 of them, and then 2 protons. That's the reaction there to make the water. And that's the water, that's the waste product of respiration, that you're making that here. If you don't remove these low energy electrons, then they'll start to clog up your whole system, and your whole system gets backed up. So in order for the electron transport systems to work properly, then you must get rid of the low energy electrons. And that's why this process is called oxidative phosphorylation, okay? Because as you're making the ATPs, that's your phosphorylation bit, you require oxygen to remove these low energy electrons. And where does the electron transport system, where are these systems? Remember we said they were on the infoldings, the cristae of the mitochondria. And remember we talked about the infoldings, that increases surface area, so you can fit more of these systems because the cristae are folded in like that, increases the surface area to fit more systems, okay? Again, go back now to your notes and look at your table there. What do you need? What are the requirements for the electron transport system? Well, you definitely need NADH, you need oxygen, fate of the products, that sort of thing, okay?